it's actually, in some sense, some of these initial experiments would be easy to do. Relatively speaking, as far as imaging goes, the visual cortex is a very imageable part of the brain, and you could do pretty basic experiments where you're passively administering the drug and just watching what happens under very sort of basic conditions that people know how to image under.
Yeah, and I would say the visual cortex is a well-studied area in neuroscience. We know a lot in terms of how the brain and the neurons there represent different kinds of visual information, and in terms of different objects, different contrasts, different oriented stimulus. I think there's quite a lot of good neurobiological questions that can be asked. In fact, I mean, the people are, getting into there. I mean, my lab, we have a couple of people working along those lines. One of the more interesting project that come online was there's also a initiative at the Allen Institute called the Open Scope, where they would perform an experiment to anybody who want to propose it. Anybody can propose an experiment, and then they would do this experiment for you. And then the condition is that you can analyze the data first, but then the data will become public a year later. So in fact, the very last open scope competition, there was a proposal to suggest that maybe we should record the visual cortex when you give animals the drug. So very soon we'll actually have a very solid, publicly available data set of how a mouse electrical activity would change
Hmm.
under the influence of psilocybin.
Interesting. So you mentioned this head twitch response assay that's often used to infer the possibility of a hallucinogenic effect in an animal like a mouse. It's very species specific. You see it in mice, you see it in rabbits, but you don't see it in all animals. In mice, the most commonly used model organism here, how tight is the correlation between a known psychedelic causing the head twitch response in that species? In other words, are there known psychedelics that have subjective effects in humans that don't cause the head twitch response, or things that aren't strongly psychoactive in humans that do cause the twitch response in mice?
Uh, in this sense, well, first of all, let me say the relationship is quite tight. So there was a very nice study comparing 25 or so different compounds that's been measured in terms of head-to-head response. And then we also know the potency in human in terms of eliciting hallucination. And then if you plot it, just, the human on one axis, rodent on the other axis, looks pretty tight. So I think that's one reason why people use it. That being said, there are a couple of false positives. So there do exist compounds where, for example, I think tryptophan is one where it's a precursor to serotonin. You give it to a rodent, then it could cause head-to-head response, but it's not, for example, a psychedelic in human. So there
Mm-hmm.
are cases like that. Whether that's owing to some, different metabolism and metabolite coming online in the rodent is unclear. I mean, there's also these things are a little hard to tell in terms of false positive because to some extent, maybe in human too, if you give enough dose at some
Yeah,
point, maybe it also become psychedelic.
yeah, yeah.
So I think it's a little hard to tell, but there are cases, a few exceptions where could be false positive or false negatives.
Mhm. So you mentioned before there's some debate out there. There's some mixed data in different systems, in culture, in vivo. Give a drug, give a different drug. There's mixed data out there in terms of whether or not the 5-2A receptor is dispensable for the neuroplasticity. What's so there's arguments that some people make that we really think it's likely that you can dissociate the subjective effects from the therapeutic effects and or the neuroplastic effects. Other people argue in the other direction. It's certainly not settled yet. One of the arguments that people use in favor of the subjective effects being important here is that when you do the human studies, you tend to find a pretty good correlation between the therapeutic effect that people have and the intensity of the experience that they report. In other words, people that report a stronger trip basically tend to have stronger or longer lasting therapeutic benefits. So this question is not resolved, but do you have a sense of how plausible you think it is that we can dissociate the subjective effects from the therapeutic effects? And whether or not you have a strong opinion there, what are the experiments going to look like that actually resolve that for us definitively?
Yeah, I have a couple of things to say about that. I mean, one is the yeah, there is human data, like you said, that suggests the intensity of that subjective effect correlate with the therapeutic outcome. To me, that is a correlation, and there could be some other factor. For example, the intensity of subjective effect would necessarily be large if the dose is also larger. I mean, if the drug response in a person is larger, which then could correlate with the therapeutic outcome. So I think these two things correlate, but I think ultimately they also correlate with the dose of the drug. Um, and then so I think it's a little hard to tease that apart. And then the other thing I will say though is that even though there has been controversy in the past, right, in terms of whether the 2A receptor is needed or not, yeah, I was actually trying to get to it, but given the past result, we have some recent result where we thought we done a slightly more precise approach to test this, ask the same question, where we also now genetically deleted the 2A receptor, but we were able to be a bit more precise and only delete it in adult animals, so not affecting development, but also deleting only in specific brain area and also in specific cell type. And when we do these very precise deletions, so not really affecting the rest of the brain or the body, um, we find that the 2A receptor is needed for some of the behavioral benefit as well as the structural plasticity remodeling.
Hmm.
Yeah, so I think, yeah, this more recent evidence kind of pushed me back towards saying that, yes, maybe the 2A receptor ultimately is also the key driver for some of the positive effects of the drug.
I see. So sort of the links and the logic here, just so people are following.
Yeah.
You give psychedelics to human beings like psilocybin. Obviously, they cause a trip. We know that happens. If you give those drugs to humans, but you block the 2A receptor, it tends to block the trip. When you give these drugs to animals, they have like the head twitch and they do the things that correlate with the hallucinogenic levels that you see in humans. And you see these neuroplastic changes. So psilocybin leads to this dendritic spine growth. It gives a head twitch response, which we think might mean it's hallucinogenic in the mouse as well. And what you're saying is there's been experiments in the past that are somewhat difficult to interpret. You can knock out or block the 2A receptor, but there's problems with those experiments. When you knock it out from birth in the whole animal, there's going to be a lot of developmental compensatory changes that make it difficult to really interpret those experiments. When you use a drug like ketanserin to block the 2A receptor in an adult, it does block the 2A receptor, but it probably blocks other things, or it might not get in the brain all the way. And it's just not a perfect experiment. You guys did an experiment where you knock out the 2A receptor only in adult animals. So you're not messing with development, and only in a certain cell type in certain parts of the brain. And what you're saying is when you give psilocybin in those specific knockout animals for the 5-HT2A receptor, it does block the neuroplastic spine growth that psilocybin would otherwise induce.
Yes, exactly. Yeah. And I think because the genetic deletion strategy that we took was just a lot more precise and also limited to adult animal, it got rid of some of the caveats on some of the early study. And I think that the effect that we saw, comparing the animals that still have the intact 2A receptor versus the animal that just have no, absolutely zero amount of 2A receptor, it's just such a night and day. When they have no 2A receptor, there's basically zero structural neuroplasticity. So comparing that differences really highlight the necessity for the 2A receptor in terms of mediating some of these long-term changes.
Okay, so the effect was strong. You basically, the plasticity that you saw with the 2A receptor intact almost completely went away without the 2A receptor. It could have been very different, right? People could have argued, well, maybe the 2A receptor is part of the plasticity and the 1A receptor is part of it, and you need, X number of receptors. And, maybe if you knocked out the 2A receptor, you would see like a 10% or a 20% reduction in change. But you're saying it pretty much all went away.
It pretty much all went away, yeah. And I think the genetic strategy have this advantage where when we knock out the receptor is no longer in the genome, and so the knockout is very complete. So meaning the receptor is practically gone.
Yeah, yeah, yeah.
Yeah.